Hello, I am Brooke.
I am Amaya.
I'm Marcelon.
And I am Bukyle.
We are four sophomores in the molecular diagnostics stream in the FIRE program at UMD. Our group is focusing on a research project on aptamer binding affinity, and we will be presenting our website at the FIRE Summit on November 16, 2020. In this video, we will introduce you to our project and walk you through the different pages of our website.
Our stream is currently working on a project where we are trying to find good aptamer candidates that bind well with a certain protein in the body. To understand more about our project, let's first talk about aptamers. In this image, you can see what an aptamer may look like once it is folded. Aptamers are single-stranded DNA or RNA segments that have the ability to bind to specific targets, which allow us to use the aptamers for diagnostics. The strength of the interaction between the aptamer and the target is measured and called the binding affinity. We are interested in finding the binding affinity of these aptamers in order to select the aptamer with high affinity and selectivity to a specific biomarker, which can then be used as a diagnostic tool.
A biomarker is a protein that attaches specifically to a cancer cell as shown in the image on the screen. If we are able to detect the presence of the biomarkers, we are able to conclude that there is the potential of cancerous cells being present in the body. In our lab, we are interested in selecting aptamers specifically against the ovarian cancer biomarker HE4, which is a protein that is overly expressed in ovarian cancer patients. In order to find the aptamers with the highest binding affinity to HE4, we have found aptamer candidates, and we are working in collaboration with Dr. Whalen's lab at the University of Notre Dame to test our aptamers with binding affinity assays. With the results from the tests, we hope to be able to create a biosensor with the best aptamer, which will then be incorporated into a screening process for ovarian cancer diagnosis.
Last year, our lab found RNA aptamers against the HE4 protein that we are planning to test using binding affinity assays. To find our aptamer candidates, we use a process called SELECTS that uses single-stranded DNA or RNA to find possible aptamer candidates for a target protein. In this image, you can see the process of SELECTS, where in this case, the DNA is first added to the target protein, and then the unbound DNA is removed from the sample. The bound DNA is then eluted from the target protein, leaving just the aptamer candidates, which are then amplified using a process like polymerase chain reaction or PCR. This process is repeated many times until the aptamer candidates are ready to go through sequencing to analyze their folding mechanisms. After sequencing, the aptamers are tested with binding affinity assays, which allows you to test their strength of the bond between the aptamers and the target protein.
One objective of our project is to develop a guide on different binding affinity assays for our lab. We hope to use it as a reference source for when we do binding affinity assays for the HE4 project and for future projects in our lab. Scientifically, we hope to ultimately use these high affinity aptamers in biosensors, which will release a signal when the aptamer binds to the target. The biosensor can be created in our lab in the future to potentially be used as a diagnostic method for ovarian cancer. The current diagnostic method for ovarian cancer is discussed in the Eaton paper written by Dr. Whalen's lab. The diagnostic method uses the CA-125 biomarker, which is currently the clinical gold standard. If we are able to use the HE4 and the CA-125 in combination, diagnosis for ovarian cancer can be more accurate. The Whalen Lab website can be seen here for more information.
On our website, you'll find pages about different topics and methods, as well as research that we have been working on in the lab. In the first topic page, the different real-life applications are described in further detail, such as the various diagnostics, therapeutic, and drug delivery methods that aptamers can be used for. In the second topic page, you can learn more about what binding affinity is and how it is measured through what is called the KD value, or dissociation constant. Binding affinity assays such as gel shift, fluorescence anisotropy, capillary electrophoresis, and surface plasmon resonance are described throughout the topic pages as well. Lastly, you'll learn more about the work we're doing in our lab, particularly related to the HE4 protein in the fourth intro page.
To learn more about the methods that we are using in the HE4 project, we are analyzing datasets from a previous project that worked with the target protein thrombin. To familiarize ourselves with the two binding affinity assays, fluorescence anisotropy and affinity probe capillary electrophoresis, we used the results from the Whalen lab in the Mears paper. The left figure shows the setup of an affinity probe capillary electrophoresis assay, and the right shows an overview of the fluorescence anisotropy assay. We describe how to interpret the data and graphs from these assays as discussed in the thrombin research paper and will ultimately apply these interpretation skills to the data for the AG4 project. Overall, in the methods pages, you can learn how the binding assays work as well as how to analyze the produced data sets. Several related research papers are included as well for more information.
In our research page, you will first find information on our past research project where we found Aptamer candidates for the HE4 project. You will then find the analysis of the data that was collected from the binding curve results of the binding affinity assays in the Mears paper for thrombin. The last component on the research page reviews the possible applications and future research projects of aptamer binding affinities. We are beginning to consider other ovarian cancer biomarker proteins that we can find aptamers against, and may even branch out to find other biomarkers for other types of cancer.
On our website, under the Additional Info tab, you can find a resources page with the sources that we used, as well as frequently asked questions page, and the team info page with pictures of our team and our majors. At the bottom of these pages, there's a link to a Google form for any additional questions or feedback.
Thank you for watching this video. We hope you were able to learn a little about aptamers and binding affinity, as well as the methods of measuring binding affinity and how we're able to use them in our own lab. Take a look through our website for more information, and please send us any questions that you may have.